So I'd like to thank the IPPN for this opportunity to introduce our relatively new phenotyping facility that we're calling the Advanced Plant Growth Centre. This has been probably 6 or 8 years in the making, and I think about 9 months or so ago, we had PSI colleagues come in and train us on its use, and since then we've been playing with it. So what I'll do really is I'll give you an overview of what the Advanced Plant Growth Centre is about, and also just give you a flavour of some of the research that we're doing, but it's very much work in progress, so please don't expect any finalised data sets, but hopefully I'll give you an idea of what we're up to. So this was funded through regional development money, actually, in Scotland, in the Tay Cities region. So that's Dundee, it's Perth, and a few cities around the River Tay, a famous salmon river. And so what we had to do was we had to demonstrate to those funding bodies that what we would do is would have some sort of economic impact. So we sold this really by saying that what we would do is we'd use next generation pre and post-harvest environments, we'd combine that with high throughput imaging technologies, would deliver underpinning science that leads to crop varieties and also supports precision and controlled environment agriculture. The Scottish government spent a while very, very interested in CEA. I think the interest has died off a little now, but there's still quite a lot of interest there. So this was really what we were all about. And what we wanted to be able to do was really, first of all, safeguard conventional crop production. We know, and we've heard many presentations over the last 2 days around how the environment's changing and producing crops is getting more and more challenging. And so we need to address those problems. And we also wanted to think about how using these sort of technologies might help us to support these new production technologies. So the questions we were asking ourselves is, how will predicted climates influence crop productivity? What's the future going to look like? We need to be prepared for that and understand what's going to happen. Can we mitigate for those climate changes by accelerating the breeding of new climate resilient crops as the environment changes ever more quickly? We need to accelerate those breeding cycles and get those new crops out into the field more quickly. What's going to happen to pathogen responses? Clearly pathogen ranges will change as the climate changes, but also we know that the abiotic environment influences how plants react with the biotic environment. And so can we understand how those changes to the abiotic environment might change plant resistance or susceptibility to current pathogens? And can we understand what's going on in natural ecosystems? Because those ecosystem services that those natural ecosystems provide are essential for farming and agriculture. And we're just waiting to hear whether we've got some work where maybe phenotyping mosses for peatland restoration in our system. And if that happens, I'll maybe need to speak to you about how we might go about that. Can we using these very high quality images and these high quality imaging environments, can we identify features which we might then be able to extend out into precision agriculture to allow people in the field to use lower cost instrumentation to allow them to understand what's going on in their field and deliver appropriate management regimes? And then new farming technologies, indoor agriculture and vertical farming being an obvious one. And we're also looking at alternative crops. We recently received a license from the Home Office to work with medicinal cannabis. And so there's quite a lot of medicinal cannabis research going on at the Institute as well. So it's built around 4 core facilities. This vertical growth tower we have a small tower and this full-size tower that contains 6 fully operated towers. They're 12 meters tall. The trays move down on lifts so that you can access them at ground level. That's a commercial company that's working with us on site and owns that. We have this post-harvest facility, but what I'm going to talk about mostly today are the next-gen controlled environments and the high-throughput phenotyping platform. And we had, again, as I say, because this was regional development funding, we have to ensure that we're interacting with industry, that we're providing those training and skills. And our ambition, if you like, was really to become this global hub for precision agriculture and draw people into Tayside. So just a very brief overview. So these were the facilities built by PSI. What we have are 8 standalone growth rooms, 4 growth rooms, which are these, what we call in the highlight growth rooms. So we can achieve up to about 1800 micromoles in there. And we have 5 separate channels of LEDs running from sort of cool white through to far red to allow us to simulate dawn and dusk and all these types of things, different latitudes. We've got CO2 enrichment there because obviously one of the key things that's going to happen is CO2 concentrations are going to go up. We need to know how that occurs. And then we also have 4 rooms where we have some additional spectra available, UVB, and I think, yeah, some of the cyans and the greens sit in there. We have a small pilot scale phenotyping platform, which we received in 2023. This was really, really useful for us because we had a good sort of 18 months, 2 years of working with this. This just takes 5 trays, which we grow elsewhere, we bring to the platform. It's semi-automated, but the software is absolutely identical to the software that we have on the large platform. And having the capacity to work with this platform and understand the software and really get to understand how phenotyping might help our research was really valuable to us. Then we now, as I say, recently developed this very large-scale platform. So this was a much, much larger thing. So what we have here are 4 growth chambers. Oops, sorry, I've moved back. We have these 4 growth chambers, which sit here. These are all independently regulated. They're identical to our standalone growth chambers. So we can achieve highlight, we can achieve CO2 enrichment, and we can manipulate both the light spectrum and the day length, etc. Plants sit on these conveyors. Each chamber takes 52 trays, and sorry, I've gone the wrong way. And the trays then feed through into our imaging loop, which has a light curtain first of all to measure the plant height, to automatically set the height of the cameras, and then we move straight into a dark and light adaptation tunnel, because if you've got 208 trays and you have to adapt in situ, you're never going to be able to phenotype in a single day. And then we move into our 3 imaging boxes. So we have chlorophyll fluorescence, RGB imaging, BNIR, SWIR, and then we have our weighing, watering, nutrition unit. As we've heard around some other facilities, we have 2 independent nutrition tanks, but we can also just grow the plants in a very rich media and provide just distilled water to them and measure water use. This is just some images of the room. So this is an example of one of our rooms. You can see these 2 automated doors at the back. So this is a plant moving out into the imaging loop, and this is the return loop where the plants then come back into the room. This is an example of or an image of the main imaging loop. So you see the 4 rooms along the side here. The plants then move out, and you've got what apparently looks like a load of dead space at the back. That's where our adaptation tunnel sits, and then you can see the 3 imaging boxes towards the front. This again is just some more detail. So these are plants coming out of the hyperspectral imaging unit, our weighing watering unit, and again returning back to the rooms. As I say, those 4 rooms can be operated independently, so we can put different conditions for a small number of genotypes, or we can operate them under the same conditions with 208 trays and different configurations in those trays. We can get up to 4,000 plants into this system, so we can do genetics as well as small scale studies. One of the issues that we're starting to encounter, and we've heard a lot about this today, and I'm sure the parallel workshop will be discussing this problem right now, is around data. So this was when we had the what we're calling the pilot scale platform. You can see we were slowly accumulating data. We then received the big platform, and within a few months we're now up to 8 terabytes of disk usage. So and really we're still not fully operational. We're still building up. So to deal with that data issue, Fraser has been developing various techniques for taking the data from the PSI server. We're very fortunate in that we have a high performance cluster available to us at the institute, just down the corridor from where the system sits. So we're developing, or Fraser has been developing, methods for really taking this database, replicating it in this Linux environment, and developing various processes for analysis of the data. And we heard about some of these. So we're using things like Dino and segment anything. So just to give you an idea of how we're using segment anything, one of the issues we have when we're growing these larger plants, such as potatoes, is they grow up, and if we want to utilize the maximum capacity of the facility, they will start to coincide with each other. But by using this segment anything, which is really a video tracking method and following things over time, it does help us to identify those individual plants and to maintain, to really segment those plants out and get pretty good quality data without having to generate a lot of our own code. An example of how we've been using that is we're trying to understand waterlogging resilience in potato. And so what we've been doing here is we've been limiting the number of genotypes that we can grow, or the number of trays that we can use. So we worked with 60 genotypes. We're imaging using RGB and various other sensors. And you can see here that using this segmenting the plants out, we actually get quite nice growth curves. And we can see when we apply waterlogging, the potatoes almost immediately stop. And when we remove that waterlogging, we can see them growing again. So this seems to work quite well. And it's a pretty quick method, actually, for getting that plant segmentation. Some other examples of some of the work that we've been doing. I have a PhD student who's been looking at the impact of elevated CO2 on barley growth. So we know from various IPCC projections that we expect, if you go to very pessimistic, by the end of the century, we're all dead because we're looking at about 1,100 ppm CO2. But some of these more optimistic projections, we could be around 600 to 800 ppm CO2. So we chose a level of 650, and we wanted to understand how that might impact barley growth and barley quality. Because if you read the literature, so this is some work done by Liz Ainsworth and Steve Long, published in 2021, and this was a massive meta-analysis of free air CO2 enrichment data. And if you look at this data, then free air CO2 enrichment should provide yield benefits, particularly for C3 crops. Although one of the concerns is that actually quality may fall. So these are just mineral nutrients, but also proteins might be expected to fall in some of those crops. So we're interested in I'm just going to show data at the moment around what we found around yield improvements. So what we did was, we're very fortunate, and we have this, what we call the barn population, available to us at the Institute. And this is a population that has, I think it's about 200 different genotypes, which are historic European two-row string barleys registered between 1830 and 2014. And the first thing the student did, Ben Potts, was he looked at the genotyping data that we've got in this group, and chose a selection of genotypes that represent that broad genetic variation that we've got within the population. And he chose 12 genotypes to grow under ambient and elevated CO2. I'm going to show you data from 4 of those genotypes, which is quite representative of the spread of data that we're seeing. And it's work that we'll be doing further analysis on these 4 genotypes. But the first thing we did was we grew them under ambient and elevated CO2, and we simply looked at the side view pixel area. And the first thing you'll observe is, in some genotypes, we saw very, very little difference between growth curves under ambient or elevated CO2. And actually, in other genotypes, they seem to do slightly better in ambient, in elevated CO2. So as you can imagine, when I saw this data, I was getting quite concerned. And I was going, are you sure you've got to put them in the right rooms? Are you sure you haven't swapped these around? And we only grew these to about 60 days. So we then moved them back into the standalone CE rooms. We grew them onto maturity, and we destructively harvested. And what we found at destructive harvest was a lot more aligned with what you see in the literature, was that we had in the elevated CO2, when we were measuring the above ground biomass, this is minus the grain, we did see this CO2 enrichment effect in all 4 genotypes. And we also saw this massive increase in the number of tillers, and actually some of the older genotypes, Golden Promise and Velvet, were possibly more affected than some of these newer genotypes. So we thought, okay, well, that's very interesting. But in 2 of the genotypes, we saw this massive reduction in grain yield, overall grain yield, despite the higher tiller number, although 1,000 grain weight wasn't impacted. And when we actually looked at what was going on in these 2 genotypes where we saw this massive reduction in grain yield, Golden Promise and Velvet, under ambient CO2, so these were all of the spikes were isolated and photographed. We got quite nice spikes. They were mostly filled, and there were quite a large number of them. But under the elevated CO2, we saw many fewer spikes, and you can also, I hope you can see, if it's good enough resolution, that there was quite a lot of grain abortion in those spikes as well. The other 2 genotypes weren't so badly impacted. And we've actually gone back and looked at the data again, and we think what's going on is that the elevated CO2 is causing this delayed development. So because we're seeing delayed development, we're getting less apical dominance, which is leading to a higher number of tillers. And we think we're getting source to sink imbalances, which is what's leading to this grain abortion and this yield loss. But that needs further analysis. But it's quite concerning that it's not seeing, we're not seeing what we're seeing in face. And again, there are probably reasons for that. I have some ideas around why that might be. And this may actually be a more accurate representation of what might occur out in the environment. Another example of some work we're doing is directly related to indoor agriculture. And what we're trying to understand is and identify markers for light use efficiency and biomass accumulation under controlled environment conditions. And the reason that we're looking for that is if you're growing plants outside, you know, they're going to have quite a high light intensity between 0 and 2000. You're going to have quite variable light. And possibly because plants have evolved in this very high light environment, photosynthesis is not particularly efficient. And actually, if you look at the amount of sunlight that's converted into biomass, we see losses all the way through. So with sunlight, about half of the energy that you get is lost because it's outside of par. We can solve that inside indoor agriculture, but many of these other elements, reflection and transmission, photochemical inefficiency, photorespiration, etc., we can't. Well, we could solve through engineering, but also I think through breeding we could get closer to this theoretical maximum than we're currently seeing. But the problem is that you're looking at about a 5% conversion efficiency of sunlight to biomass. So the question that we asked ourselves was, in indoor farms, well, can we identify genetic variation which might help us to get closer to this theoretical maximum? So we were very fortunate. We were working with a breeding company in the UK that had an F7 inbred lettuce population, a cross between baby leaf green romaine and green iceberg, 176 individuals plus parents. And these just show you an example. So we've got 4 different genotypes in each of these images. Just gives you an idea of the variation that you can see within that population. The first thing that we did was working with our partners, Intelligent Growth Solutions. We grew these up in their indoor farms. You can see these are staggered because we wanted to do gas exchange work on these. We were limited in the amount of gas exchange we could do in a single day, so we had to grow them, as you can see, in this staggered way. In the beauty of working in the indoor farm, it's a very controlled environment. So even though they weren't grown at the same time, yes, there were edge effects before anyone asks, but we were able to grow them for 28 days in precisely the same environment before we then took them at that 28-day point and did our gas exchange. And we did a bunch of AI curves and ACI curves, and the good thing is we get this nice sort of normal distribution, both in sort of photochemical traits, in biochemical traits, and in CO2 assimilation. When we did the correlations, we get correlations where we'd expect to see them. So things like maximum rates of assimilation correlate very nicely with some of these biochemical traits and also with the photochemical traits. The thing that was really quite, well, not concerning, but that kind of blew my hypothesis out of the water that biomass would be limited by CO2 assimilation under these low light CE environments. We saw very little correlation between the biomass parameters and the photosynthetic parameters. So the next thing that we did was to put this population into the phenotyping platform and measure their growth rates over this 28-day period. Again, we've got this very nice sort of normal distribution in growth rates. We're currently genotyping the population, and very soon we hope to link those genotypic markers to these various traits. So that's just a very brief overview of what we're doing in the Advanced Plant Growth Centre. I'd like to finish by acknowledging all our funders and all the people who have been involved in this. So Ray and Akis are 2 postdocs in my lab who, as well as their normal duties, are now very much focused on operating this platform and making it work. Ben's the PhD student who did the barley work. Sanjeev helps us out with the genetics, and Fraser and Dominic help us with the image analysis. The whole thing was funded through this Tay Cities Region deal, which was Scottish Government and UK Government. The phenotyping platform itself, we received a very large grant from Scottish Enterprise. Much of the work's funded by the Department of Environment, Food and Rural Affairs, UK BBSRC, Innovate UK, and very shortly we'll be doing some phenotyping of legumes for feeds, and that's part of the Horizon Urine project. Thank you for listening.
All right, Rob. Thank you very much for this very nice and interesting introduction. Please stay here. Maybe there may be one or the other question to you. So the talk is over.
Sonia.
Yes, Sonia, please.
Yeah. Yes, we've got that. Yeah, but I mean, it's not in a fit. It's not in a fit state to show just now.
For the online participants, could you repeat?
Yes. Yeah. So the question was, in the barley ECO2 experiment, did we look at chlorophyll fluorescence? The answer is yes, we did, but it's currently not really in a fit state to show. But what we did see, again, initial estimates are that actually we grew at the material that was grown at elevated CO2. Now we did the chlorophyll fluorescence at ambient CO2, and what we're seeing is actually a lower quantum yield from the stuff that was grown at ECO2. And I suspect the reason is because it's growing in that ECO2 environment. It's used to that CO2 enrichment. When we transfer it back into an ambient CO2 environment and then excite the photosystems, there's perhaps not quite, because there's perhaps not quite enough of the biochemical cycles occurring, or the biochemical cycles aren't working hard enough under ambient CO2, I think. Again, speculation. Yeah.
Yeah, Rob, thank you for the presentation. I also have a small question to the CO2 experiment with barley, because you said there was no increase of biomass in the period you observed it, and later you showed there was a way higher tiller number.
Yeah.
So were the tillers formed after the imaging period, or isn't that?
So I think, so I think what's happening is we're getting we saw a slightly delayed germination because it was, they were germinated under elevated CO2. The entire growth cycle was under elevated CO2. We saw a slightly later germination, and when you looked at the plants later on, they had a lower stature. And I think what was going on is I think we have delayed development, full stop. Because I think we've got that delayed development, I think that's allowing, because the apical dominance from the primary tiller occurs later, I think that's allowing more secondary tillers to come out and grow. And again, this is all speculation, right? And we need to do that experiment. But I think that's perhaps what's happening. Yeah.
Okay. First row first, and then yes.
So the same question for the barley experiment. Any effect on other leaf components, like the length of the tiller?
Yeah. So again, we're just analysing that data now. So the tillers were shorter. Definitely, there were more of them, and the spike, I don't know if you saw in those images, the spikes tended to be shorter, and we're getting higher grain abortion in those spikes. Fraser is not here now, has just developed an algorithm to allow us to use image processing to look at the total grain number and the aborted grain number and grain sizes in those images. We're analysing that data now, but I think we are seeing smaller spikes and more abortion within the spike, but I've not quantified it yet. That's just from looking at the data.
Okay, Carlotto?
Well, it's a challenge when you change CO2. Plants really get like that, at least the speech I think.
Yeah.
Either they've been growing on high CO2 pump. The person says that.
Yeah. Yeah, and the question is, yeah. No, it's constant elevated CO2, 650 ppm. But obviously, the plants leave those rooms, and this again, maybe we'll have these conversations later. But from our limited experience of controlled environment phenotyping, you're always making a compromise, and the compromise that we're making there is obviously the plants are leaving those rooms on it every day, and each plant is probably out of the room for 20 to 30 minutes, and then coming back into the room. So, yeah, that's a confounding effect, without a doubt.
Okay, I also have a question about the environment regimes that you control there. do you run this typical phytochamber light on, light off, or do you do temperature fluctuation?
No, at the moment we're keeping it simple, but it's not a square wave. We are ramping up and then ramping down. And we ramp the temperature along with the light as it comes up over half an hour, and we ramp down over half an hour. But then it's completely constant during the day. Yeah, we'll get to that later, no doubt.
So our experience was you need to challenge the plants and have them irregular changes from day to day and different profiles, and then they are close to what. If you do it very regularly, then they adjust to it and grow faster as usual. Okay, good. Thank you. Any further question right now to Rob? Okay, then thank you very much again.